day, learners! How are you today? We hope that you're all fine. I'm your teacher Jun Rey, and I'm here with Teacher Jaysa, Teacher Jeffrey, Teacher Jeremie. We will discuss our next topic in cell and molecular biology. Before anything else, let's have first a short prayer. Class, let me check first the attendance. I'm so glad that you're all present. You have perfect attendance. By the way, do you still remember the topic that we have discussed last time? Yes, what was our previous topic? That's right. We have discussed the genetic code and the translation. What is genetic code? Yes, genetic code is the term we use for the way that the four bases of DNA, the A, C, G, and the T's, are strung together in a way that the cellular machinery, the ribosome, can read them and turn them into a protein. In the genetic code, each three nucleotides in a row count as a triplet and code for a single amino acid. So each sequence of three codes for an amino acid, and proteins are made up of sometimes hundreds of amino acids. So the code that we would make one protein could have hundreds, sometimes even thousands, of triplets contained in it. How about the genetic translation? Correct. Translation is the process of translating the sequence of a messenger RNA or the mRNA molecule to a sequence of amino acids during protein synthesis. The genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid ribosome sequence that it encodes. In the cell cytoplasm, the ribosome reads the sequence of the mRNA in groups of three bases to assemble the protein. I have here two pictures. What do you think is in the first picture? Yes, it's a DNA or deoxyribonucleic acid. How about the second picture? That's right. Those are technologies or different form of technologies. How about we put them together, DNA and technologies? Correct. We have now the DNA technologies. What does DNA technologies mean? Okay, it means that we use technology in studying and experimenting the DNA. DNA technology is the sequencing, analyzing, and cutting and pasting of DNA. Common forms of DNA technology includes DNA extraction, polymerase chain reaction or the PCR, DNA microarray, and gel electrophoresis. In this lesson, we are going to discuss the different technologies of what are their uses and how they are being done. So listen attentively and participate actively. First is the DNA extraction. What is DNA extraction? Yes, this is a method to purify DNA by using physical and chemical methods from a sample, separating DNA from cell membrane proteins and other cellular components. As you can see in this picture, the DNA is a long, stringy molecule that can be lifted out of a solution by the use of a glass rod or wooden stick, which it naturally wraps around when turned. What are the applications of DNA extraction? The ability to extract DNA is of primary importance to studying the genetic causes of disease and for the development of diagnostics and drugs. It is also essential for carrying out forensic science, sequencing genomes, detecting bacteria and viruses in the environment, and for determining paternity. So what are the importance of DNA extraction? The extraction of DNA is pivotal to biotechnology. It is the starting point for numerous applications ranging from fundamental research to routine diagnostic and therapeutic decision-making. Extraction and purification are also essential to determine the unique characteristics of DNA, including its shape, size, and function.
Gel electrophoresis is a biotechnique used to separate DNA fragments and other macromolecules such as RNA and proteins based on their size and charge. Gel electrophoresis is a process by which we take the DNA and run an electric changes through it. Therefore, we can use it to compare two DNA samples, hence the DNA fingerprinting. The molecules to be separated are pushed by an electrical field through a gel containing small pores. The molecules travel through these pores in the gel at a speed which is inversely related. This means that a small molecule will travel a greater distance through the gel has a positive charge and the other end has a negative charge. DNA is a charged molecule, so DNA molecule will move when an electrical field is applied to a liquid in which they are dissolved. If the liquid is a simple one, such as water with some salts in it, all the DNA molecules moved at nearly the same speed. Under these conditions, it is hard to distinguish the tiny disparities in the motion of different kinds of DNA. If the solution is made less liquid as in a gel, and the DNA molecules all start moving across the solution from initial small volume that is from essentially the same starting point, then the molecules can move at perceptibly different speeds, usually smaller DNA molecules more faster than larger ones.
Good day everyone, I am Jeremie Udtohan and my topic is all about DNA microarray, the continuation of our lessons, DNA technologies. So, what is DNA microarray? The DNA microarray is a tool used to determine whether the DNA from a particular individual contains a mutation genes. What is DNA microarray used for? DNA microarray were used only as a research tool. Scientists continue today to conduct large-scale population studies, for example, to determine how often individuals with a particular mutation actually develop breast cancer, or to identify the changes in gene sequence that are most often associated with particular diseases. The DNA microarray also used to study the extent to which certain genes are turned on or off in cells and tissues. In this case, instead of isolating DNA from the samples, RNA, which is a transcript of the DNA, is isolated and measured. Today, DNA microarrays are used in clinical diagnostic tests for some disease. Sometimes, they are also used to determine which drugs might be best prescribed for particular individuals, because genes determine how our bodies handle the chemistry related to those drugs. Why DNA microarray is an important tool? DNA microarray is an effective tool in transcriptomics that helps us in studying and analyzing the mRNA expressions of almost every often present in an organism. Functions of a DNA microarray. It helps to pinpoint the functions of specific genes and to compare the expressions of genes in different types of cells, organisms, and can be used to detect genetic mutation by comparing an individual's DNA with normal DNA, often used to compare cancerous cells to regular cells, also called a genochip, because technology used is similar to that used to lay out electronic circuits on a computer chip. Structure of a DNA microarray. So it is composed of a solid surface that has a microscopic grid of many DNA fragments attached to this. These DNA samples are called probes. Probes are single-strand and represent every protein coding gene in the human genome, designed to hold between 10 to 2.1 million probes. How DNA microarray work? mRNA from two types of cell is isolated reference cell, experimental cell. Extracted mRNA used to build complementary DNA or cDNA libraries. cDNA from each cell is labeled with an specific fluorescent type. cDNA sequence denatured into single strands and pumped across the surface of the microarray. Laser detection system is used to detect fluorescent. Reading the result of microarray. Since microarray contains probes that correspond to coding genes and cDNA that is complementary to the probe sequence will bind to this probe or hybridize. cDNA with no matching sequence will be washed off the chip. No digen that expressed. i.e., if reference cDNA was originally dyed red, and experimental cDNA was originally dyed in green. So red gene expressed in control cell only, while green gene expressed in experimental cell only, while yellow gene expressed in both cell, and black gene not expressed in either cell. So practical uses of DNA microarray. Practical uses allows researchers to learn more about many different diseases including heart disease, mental illness, and infectious disease. The study of cancer, classifying types of cancer based on patterns of gene activity in tumor cells, designing treatments targeted to each specific type of cancer, examining difference in gene activity between treated and treated tumor cells to understand effect of different therapies. So that's could be all my topic, DNA microarrays. Thank you and God bless.
What is CPR? The polymerase chain reaction, or PCR in short, was developed in 1983 by American biochemist Kary Mullis. He was awarded the Nobel Prize in Chemistry in 1993 for his pioneering work. PCR used in molecular biology to make many copies of small section of DNA or genes. Using PCR, it is possible to generate thousands to millions of copies of a particular section of DNA from a very small amount of DNA. To answer how does PCR work, let's talk about what we need first before we get the steps of how it works. The first is DNA portion. We need DNA portion that we wanna make copies of. Example of a DNA portion is a genes. The second is a buffer. We need some kind of buffer to put it in. A buffer is a solution or substance that has the ability to maintain pH and bring it back to its optimal value by addition or removal of the hydrogen ions. Also, we need things that would be necessary to make more copies of a DNA. The third is a primers. It helps DNA polymerase a building enzymes to where to go to start buildings. A primer is a short single-stranded nucleic acid used by all living organisms in the initiation of a DNA synthesis. Number 4 is a DNA polymerase, a building enzyme from that a DNA polymerase use obtain a heat resistance that a type of a DNA polymerase as PCR uses heat. Typically, the polymerase chosen for the job is a Taq polymerase. It is a type of a heat resistance from a DNA polymerase. Taq polymerase is originally a type of bacteria that can handle and live in a really high temperature nature in hot spring. Last is a DNA nucleotides. We also need DNA nucleotides polymerase to build with it. Step 1 is denaturation. In this step involves the addition of heat is needed to separate the two strands of a DNA molecule. Denaturation also separates the two DNA chains by heating the test tube to 90 to 95 degrees centigrades. The step 2 is annealing. Basically, this is just when the two DNA strands has been separated by the heat are going to be cooled and be joined by the primers. The temperature for this step should allow the primers to bind to the specific segment of a DNA that you want to amplify, which means make copies of. The step 3 is a DNA synthesis. Remember, synthesis means to make something. With DNA synthesis, we're going to make more of DNA. And to do so, DNA polymerase will begin to work on both strands and it will use nucleotides as its building materials to amplify the DNA. We should know that the temperature of these steps may be a little warmer than the previous steps. Now you have two double-strand DNA molecules to start with. So you repeat with denaturation, annealing, and DNA synthesis step. Now you have 4 double-strand DNA molecules.
I guess you have now learned about DNA technologies from your teacher's discussion. To evaluate what you have learned, we have here a 10-item multiple choice test. You have 20 seconds for each item. Okay, time is up. Thank you, class, for listening to your teachers for today. We hope that you learned a lot from our discussion. Enjoy your day and God bless. Goodbye, everyone.